Well, hello everyone. My name is Yu-Qin. Well, first of all, I would like to thank Professor Lin for organizing this meeting and to all those who attended. And I'm very glad to be here to represent my professor, Dr. Bao Pen, to introduce our work, which is
Mm.
titled Thermodynamic Chemical Reaction Network Drove Autocatalytic Prebiotic Peptides Formation and the Prebiotic Chemical Evolution. And the the
Yes,
can
can
you
you?
see the next PPT?
No, we only see the first slide.
Oh, oh, it's there something wrong in my computer.
Mm.
Oh, now
Okay,
can you
yeah,
see?
we can see
Oh,
it now.
yeah.
Yeah,
Okay.
maybe it's just the slow connection. Yeah.
Yeah. And now I would like to first introduce our research direction. Our research focuses are centered around the following four aspects: probiotic chemistry, astrobiology, and sulfur geochemical cycle, and energy, e- early and energetic metabolic bacteria. And okay, now let's begin today's topic. The question of how life began is as old as human civilization itself. And for thousands of years, people have asked, where does life come from? And in modern science, this remains one of the most fundamental and challenging questions because it covers many fields, including biology, chemistry, physics, and even philosophy. As early as ancient Greece, philosophers suggested that life is inherent in matter and can arise spontaneously under suitable conditions. And however, these ideas are largely philosophical and lacked experimental evidence. So this brings us to the key question: how can life emerge from non-living matter? And Um, in modern orange of life research, the chemistry of life is often understood as autocatalytic chemical reaction networks or CRNs, which involves the combining of six key elements: carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus. And based on this idea, we propose that on early Earth, there may have existed a thermodynamically driven chemical reaction network, mainly involving carbon, hydrogen, oxygen, nitrogen, and sulfur powered by energy-releasing redox reaction, especially when phosphorus was limited. And our main focus is autocat is autocatalysis, which can be understood as a form of non-genetic self-replication and is considered as a considered and is considered a key step towards the origin of life. And from a geochemical perspective, on the early Earth, volcanic and hydrothermal activity likely produced a lot of sulfite and nitrogen mainly existed as ammonium. When sulfite, ammonia, and carbon dioxide interact, they can react through redox reaction, releasing energy and producing simple organic molecules. And we call this process Sammox, which stands for sulfur reduction compound with anaerobic ammonia oxidation. And it formed a chemical reaction network that may provide both the energy supply and the reaction pathways needed for early probiotic chemistry. And to test this idea, we set up a Sammox-driven reaction system. We mixed the sulfate, or sulfide, ammonium, and bicarbonate in a stereo system without oxygen. And the system was heated first at 100 degrees and then at 70 degrees. And this step up mimics the early Earth condition. And in this system, multiply reactions happens at the same time. Ammonium and sulfur react with each other through redox reactions, producing molecules like nitrogen gas and hydrogen sulfide. Importantly, small organic molecules such as fomate are formed, and all these reactions are linked together, forming a chemical network. Well, let's see what happened in the first experiment. We found that both the sulfate and the sulfite-driven system produced small amount of fomate and a higher amount of peptides. And however, um, in the sulfite system, only a little fomate was formed and almost no peptide were detected. And this suggested that the reaction may need higher temperature or a catalyzed. Overall, we think the fomate is the first product in the network, while peptide are the final products, especially in the sulfite and sulfate-driven system. And to analyze the reaction product, we used chromatographic to detect if peptide was formed with detection at 214 nanometers. And we found a small pack around 2.6 minutes. And then we use mass to find out the composition of this peptide. And for the mass results, we found that these peptides are made of 15 amino acids. And this is important because it stressed that complex life-related molecules can form from simple inorganic substance under suitable conditions. And if we look at the reaction network more clearly, we can see that it is not a simple linear pathway, but a highly interconnected system. Well, for example, the carbon dioxide is first reduced to form small organic molecules such as fomate, which then act as key intermediates. And these intermediates can be further converted into amino acids and finally into peptides. And this shows a clear pathway from simple inorganic molecules to more complex life-related compounds. And to further study this process, we carried out experiments on reductive ammoniation. We reacted ammonia fomate with different keto acids, and this system was heated at 70 degrees for 48 hours. Well, we found that different amino acids were produced in all cases, and although the types varied depending on the starting material. And this shows that Sammox-driven chemistry can produce a variety of amino acids, which is an important step toward building more complex biological molecules. And in the third experiment, we wanted to check if the Sammox system has self-catalytic behavior. So we designed a two-stage experiment. Well, in the first stage, we let the reaction happens as usual, like experiment 1. And then we took some of the first stage products and add them into a fresh system and to see if they could help drive the next reaction. And also, the next reaction was heated at 100 degree for 24 hours and 70 degree for 24 hours. And after analyzing the products from the second stage reaction, we observed an interesting pattern. The products generated from the sulfite-driven Sammox system in the first stage were able to promote peptide formation in the sulfite-driven system during the second stage. But at the same time, they inhibited peptide formation in the sulfate-driven system. And on the other hand, the products from the sulfate-driven Sammox system showed the opposite effect. They promoted peptide formation in the sulfate-driven system while inhibiting the sulfite-driven system. And this indicates that the reaction products can affect different systems in different way. And also, we repeated the same experiment at a lower temperature of 45 degrees for 48 hours. And interestingly, the results showed an opposite trend in peptide concentration compared to the higher temperature conditions. And this suggested that temperature plays a critical role in controlling how the system behaves. And overall, this result showed that the peptide formed in the Sammox system can either promote or inhibit reactions, and depending on the conditions such as temperature and the chemical environment.
Sorry, I have a quick question. What is the F and R here?
Well, for example, F means the product in the in the sulfite in the sulfite can also promote the sulfite in the second stage, but and the R it means it will inhibit the second stage. So they are opposite in different temperature and different stage.
Thank you.
Yeah, and this opposite behavior can be described as a seesaw-like effect. And in the other word, peptide produced in the sulfite-driven and sulfate-driven Sammox system have opposite role in controlling the reactions. And this two-way effect helps the system keep a balance between building up molecules and breaking them down. And similar to what happens in our living systems. And because of this, the seesaw-like behavior may help keep the whole reaction network stable and able to adapt to different conditions. And well, in the first experiment, we aim to investigate whether the peptide products in the Sammox system could actually promote chemical reaction. So we focus on two process, the sulfite-driven Sammox reaction and anaerobic ammonia oxidation. And we added the peptide into this system to see if they could make the reaction go faster or more effectively. If they could, it would suggest that this peptide have enzyme-like or early catalytic functions. And the results show that peptide from the sulfite-driven Sammox system significantly promoted the reaction, and they increased the concentration of sulfite and ammonium in the Sammox process, and also enhanced the concentration of nitrate and ammonium in anaerobic ammonia oxidation. And these findings stress that the peptides are not only products of the reaction network, but also active components that help drive the reaction. And therefore, we conclude that the Sammox-driven reaction system exhibited a proto-metabolic potential as it demonstrated both catalytic activity and network level function network level functionality. And to further investigate the evolution of this system, we carried out a long-term experiment. We kept the reaction system under normal conditions for up to 3 years, allowing slow reactions and environmental change to influence the system. And after long-term incubation, we were surprised to detect nitrate in the Sammox reaction system. And since the experiments were conducted under sterile conditions, this observation stressed that the nitrate was produced through abiotic or proto-metabolic processes rather than microbial activity. And this phenomenon is constant with an anaerobic ammonia oxidation to nitrate pathway, and sometimes referred to as an AAON-like process, which has been reported in Sammox-driven chemical system. And the formation of nitrates indicate that the reaction network may have developed more complex nitrate nitrogen transformation over time. And in addition to these chemical changes, we also observed a stable vesicle-like structure under microscopic. And these vesicles are significant because they can provide compartmentalization, which is a key function of living system. And their formation suggested that the Sammox-driven chemical system may not only support molecular complexity, but also began to develop a special organization, which is a step towards cell-like structure. And based on all these findings, we propose that probiotic evolution progresses from redox reaction to chemical reaction network, and ultimately to biochemical network. And the phenomena of active peptide catalytic function evolution occurred in the Sammox-driven chemical reaction network. And this is all the content, and thank you for your time.
Thank you.